Okay, so let's get started. Hello, everyone. Welcome to our today's Zoom seminar. It's really my pleasure to welcome our today's speaker, Dr. Daichao from Chinese Academy of Science Interdisciplinary Research Center on Biology and Chemistry. So, um, Daichao actually graduate his undergraduate training in Zhejiang University and then continue his PhD training in Shanghai Institute of Organic Chemistry, Chinese Academy of Science. um, and then after that, he moved to U.S. To Harvard Medical School, complete his postdoc training, and then after a relatively short postdoc training from 2019, he moved back to China, start his independent research program in the interdisciplinary research center of biology and chemistry, CNS. The research in Daichao's lab mainly investigated proteins which can regulate cell death and inflammation. His lab is revealing how these proteins contribute to aging-related disorders, inflammatory and then degenerated diseases such as premature aging, Alzheimer's disease, as well as pathogen infections. Um, the main goal of his lab is to translate these discoveries into new treatment for these sources. And then there are several different aspects Daichao's lab actually mainly interested in, including discovery of programmed cell death checkpoints and the molecular machinery by which their inactivation leads to human disease. And the second part is biochemical studies of key proteins in inflammatory cell death pathways and their physiological and pathological functions. The third part of his research is to discover novel cell death pathways and their pathological functions. Last but not least, his lab also studying molecular machinery underlying the progression from aging to neurodegenerative disease and drug development. I think I was reading through Daichao's CV. Um, Despite he just being back for to start his independent research for short like 6 years, but he's been extremely productive. I was completely blown away. And then with all these achievements, he was rewarded by different honors, which I'm not going to list them all, but several highlights, including science exploration scholars, National Science Funds for excellent young scholars, and then National Science Funds for distinguished young scholars. So it's really pleasure to have Daichao with us today, and then we are very much looking forward to our talk.
Okay, so first, thank you, Tran, for the invitation, and I'm happy to be here on in this online seminar to present our work. And today, I would like to share a very recently published story about the role of a protein called RIPK1, which is a cell death protein that is we are studying for many years, as have a new function where it has can act as a sensor of a very important metabolite called S-adenosylmethionine, and where RIPK1 can detect the scarcity to drive cell death and inflammation. So a fundamental question in metabolism is how cells sense nutrient to maintain cellular homeostasis. And we know that the capacity of cells to sense and respond to changes in nutrient levels is essential for their survival. But previous studies have been focused for a long time for identifying nutrient sensors in the metabolic pathways. But the mechanisms that links nutrient sensoring to the cell death pathways, such as apoptosis and necroptosis, are, are not well defined. So, for example, amino acid are bioactive molecules that is essential for not only metabolic homeostasis, but also preventing cell death, because more than 20 years ago, studies can found that deficiency in several amino acids, such as methionine, can induce apoptosis. But the mechanism by which they regulate cell death remain unclear. So what is RIPK1? So RIPK1 is a key mediator of both cell death and inflammation, and it is most studied in the TNF pathway. So upon the TNF stimulation, RIPK1 interact with TNF receptor 1, and this will induce a complex called complex 1. And so the purpose of this formation of this complex 1 is to induce the activation of NF-kappa B pathway that is required for cell survival and for pro-inflammatory responses. And at the same time, so there are many. So RIPK1 is also a kinase, and in normal conditions, the kinase activity of RIPK1 is suppressed by many a complex set of post-translational modifications. And so far, we know that dysregulation of each of these modifications can lead into the kinase activation of RIPK1, leading to its autophosphorylation and interaction with the protein called FAT to form complex 2, and which drives caspase-8 mediated apoptosis. And alternatively, RIPK1 upon activation can interact with RIPK3 to form a complex called necrosome. And this will finally activate MLKL that execute necroptosis. So in addition to cell death, RIPK1 kinase can also drive inflammation. So this can happen in both cell death dependent and cell death independent manners. Okay, so importantly, mutations in genes that encodes these proteins that inhibit RIPK1 kinase activity can lead to diverse inflammatory and degenerative diseases, and which are believed to be caused at least part by the overactivation of the kinase RIPK1. Okay, so given the central role of RIPK1 kinase in mediating cell death, we screened wild-type MAFs and the RIPK1 D130N kinase dead MAFs for cell death in response to various amino acid deprivation. So prolonged deprivation of methionine, cysteine, and glutamine for like 72 hours induced a significant cell death in wild-type cells. But interestingly, we found that the cell death induced by methionine deprivation was specifically blocked by RIPK1 kinase inhibition. So this suggests that RIPK1 kinase drives the cell death induced by methionine deprivation. So as expected, because RIPK1 kinase drives the cell death, we can see that methionine deprivation indeed induced the kinase activation of RIPK1 as marked by the S166 phosphorylation, which starts from like 12 hours after methionine deprivation and peaked around 72 hours. And the methionine deprivation also induced apoptosis, just like the report 20 years ago. And this was marked by the caspase 3 cleavage, or CC3, and the cleavage apoptosis phenotype was totally blocked by the RIPK1 kinase dead mutation. But in contrast, necroptosis is not involved because knockout of MLKL, which is the executor of necroptosis, was ineffective to block the cell death. But deficiency in FAT, which we just said it is required for the activation of caspase-8 and apoptosis, blocked the cell death. So this further confirms the involvement of apoptosis in the cell death induced by methionine deprivation, but not necroptosis. And we just said before, RIPK1 kinase can also induce inflammation in addition to cell death. So we performed RNA-seq analysis of the methionine depleted cells, and we found that the methionine deprivation induced a significant upregulation of genes in wild-type MAFs. And we can see that more than 1/3 of these genes can be reversed to like normal levels by RIPK1 kinase inhibition. Okay, and so we can see that these reversed genes are predominantly associated with inflammation and innate immunity, which suggests that methionine deprivation also induces RIPK1 kinase mediated inflammation in addition to apoptosis. So this is important because we usually think about that we usually think the apoptosis is not inflammatory cell death, right? But in this case, because of the kinase activation of RIPK1, it can also drive inflammation. Even the cell death modality is apoptosis. Okay, so we also studied the physiological relevance in vivo, and we subjected the mice to a methionine restriction diet that contains 0.12 methionine for 8 weeks. And consistently, the wild-type mice exhibited increased expression of inflammatory genes in the liver. And while the RIPK1 kinase dead, kinase mutation can completely block this. And we also observed the liver damage in wild-type mice, as determined by the increased levels of serum ALT and AST, which were also restored to normal in RIPK1 kinase dead mice. Okay, so this result suggests that methionine insufficiency can promote RIPK1 kinase activation, leading to both apoptosis and inflammation. But how this happens? So methionine is essential for the biosynthesis of S-adenosylmethionine, or SAM. So SAM is a key component of the one-carbon metabolism, which compromises the methionine and the folate cycles that deliver one carbon unit to various biosynthetic pathways. And interestingly, we found that only methionine and the SAM supplementation can inhibit the cell death, but other metabolites from the cycles were ineffective. Okay, so what is SAM? So SAM is a co-substrate involved in methyl group transfers, which donate its methyl group to diverse acceptors, including nucleic acid, proteins, lipid, and the secondary metabolite. And the conversion of methionine to SAM is mediated by the methionine adenosine transferase, or MAT. So we used the specific inhibitor FADAS5 to inhibit the MAT, and which reduced the SAM levels as expected. And interestingly, we found that MAT inhibition alone induced the cell death. And this cell death can similarly be inhibited by the supplementation of SAM, as well as by RIPK1 kinase dead mutation. Okay. So this result suggests that the generation of SAM is important for suppressing RIPK1 kinase activation and the kinase dependent cell death. And because of this reason, we predict that alterations in the one carbon flux, which is essential for SAM generation, will also promote RIPK activation. So, for example, we use an inhibitor to inhibit the an enzyme called S-adenosylmethionine in the one carbon metabolism cycle, and this inhibition can lead to the reduction of SAM levels without affecting the methionine levels. And consequently, we can see that this also induces cell death that is suppressed by the supplementation of SAM, as well as by RIPK1 kinase dead mutation. Okay. So this result suggests that the downstream generation of metabolism and the one carbon metabolism is essential for preventing RIPK1 activation. Okay, but how this happens? So as we found that methionine deprivation or neutralization of TNF using an anti-TNF antibody also inhibits the cell death. So RIPK activation depends on the canonical TNF detection pathway. So as a methyl group donor, SAM is essential for intercellular methylation reactions, including DNA methylation and the protein methylation, okay? And the DNA methylation is commonly invoked as a silencing mark that suppresses transcription. And indeed, we found that in response to methionine deprivation, the levels of TNF promoter methylation were reduced. Okay, so this indicate the increased transcriptional activation of TNF. And we think that this likely contribute to the initial autocrine production of TNF alpha that is required for promoting the cell death in methionine depleted condition. Okay, so we then used the inhibitor ADOX to suppress SAM-dependent methylation reaction. So ADOX specifically inhibits SACH hydrolyze, and this will lead into the accumulation of SACH, and SACH can further inhibit SAM-dependent methyltransferase, thus inhibit the SAM-dependent methylation reaction. So as expected, ADOX treatment substantially decreased the SAM-SACH ratio, which is an indicator of the methylation potential, without affecting the SAM levels. Okay, and again, ADOX also induce the cell death. And this results suggest that SAM-dependent methylation reaction, rather than SAM itself, suppress RIPK1 activation. So because it is well known that TNF alone is not sufficient to induce RIPK1 mediated cell death in normal condition, right? Because TNF stimulation usually induce the activation of NF-kappa B pathway for cell survival. But because there are a lot of proteins that suppress RIPK1 kinase activity to prevent cell death. Okay, so the autocrine production of TNF alpha upon SAM depletion cannot explain the cell death phenotype. Okay, so we're trying to identify additional regulatory process that is dependent on SAM-mediated methylation. And we come out with the notion that RIPK1 is usually suppressed by many distinct post-translational modifications. So we wondered maybe RIPK1 can undergo methylation by SAM. And indeed, mass spectrometry analysis revealed that there are 3 RIPK1 residues that can be dimethylated in normal conditions, but substantially reduced upon methionine deprivation. And among these 3 residues, only R606 is conserved across species. So then we focused on the dimethylation of this residue and to see whether the dimethylation of this residue contribute to the RIPK1 activation regulation. And because arginine dimethylation occurs in 2 forms, the first form, asymmetric dimethylation, which is catalyzed by the type 1 protoarginine methyltransferase PIMT, and by the type 2 PIMT. So as detailed below, because we found that this dimethylation is catalyzed by a type 2 PIMT, suggesting that this the dimethylation of R6 is symmetric dimethylation body that can specifically detect methylation or Me2S of the R606 residue. Okay, and using this antibody, we found that under normal conditions, endogenous RIPK1 is dimethylated on R606, which was reduced upon SAM, upon methionine deprivation, but can be recovered by the supplementation of SAM. And while depletion of SAM, while the inhibition of MET2MET or the SHMT inhibition or suppression of SAM-dependent methylation by ADOX, all significantly decreased RIPK1 dimethylation event. And then next, we characterized the physiological relevance of RIPK1 dimethylation in vivo. So we examined the liver tissues from mice fed a methionine restriction diet, and RIPK1 dimethylation was detected in livers from mice on a normal diet, but was absent in those on a methionine restricted diet. So this suggests that RIPK1 is dimethylated under normal physiological conditions. Okay. So next, we started with the RIPK1, R606 methylation regulate its activation and cell death. So because methylation suppress RIPK1 activation and cell death, right? So we expected that loss of methylation by the R606K mutation will enhance the cell death. But however, indeed we found that this mutation reduced the cell death, and it is also inhibited the RIPK1 activation and apoptosis. So this suggests that RIPK1, the R606 residue by itself must be involved in promoting RIPK activation. And only in this case, when this residue is methylated or mutated, it reduces RIPK activation, right? Okay, so because this mutation suppresses RIPK1 activation, we then generated a RIPK1 R606K looking mice to study how strong this mutation can suppress RIPK activation. So we used the methionine restriction model, and we performed RNA-seq analysis of liver tissues from wild type and the mutant mice, and we found that just like the RIPK1 D138 mutation restored the expression of more than one-third of genes to normal levels. Again, these genes were enriched just like the D138. So in the TINF, there is a very well-established model to study the RIPK1 kinase active function in response. And we want to see how that these R606K mutant mice showed complete resistance to TNF-induced hypoxemia and lethality. So these results together suggest that the RIPK1 R606K mutation can protect the mice from cell death and inflammation induced by methionine restriction or by TNF stimulation. So just similar to the D138 kinase dead mutation. Okay, so then the question is, how does the dimethylation of this residue suppress RIPK activation? Or we can ask, how does the R606 residue by itself promote RIPK activation? Okay, so the R606 residue is located in the death domain or DD of RIPK1. So DD-mediated dimerization of RIPK1 is known to be essential for RIPK1 autophosphorylation and the kinase activation. Okay, so then we test whether this residue is involved in RIPK1 dimethylation, and indeed we found that the R606K mutation decreased the self association or dimerization of the RIPK1 DD. Okay, so RIPK1 DD protein is prone to form oligomers through self-association. So during the purification, the DDR606K mutant yield more monomers and less oligomers than the DDY type protein. Okay, and we found that the DDY type monomers tend to form a long filament over time, but in contrast, the DDR606K mutant show the less and shorter filament compared to DDY type, which is consistent with the reduced self-association of the R606K mutant. So to elucidate the molecular details, we solved the cryo-EM structure of the DD filament. So from the structure, we can see that the side chain of R606 is directly involved in the DDDD interaction interface. So basically, we can see that the R606 of one DD subunit forms interactions with E605 from another DD subunit. So thus, we can expect that disrupted by mutating the arginine to lysine. Because the side chain of arginine is more extended than that of lysine, this allows for closer interaction with the E605. And in addition, the hydrogen bond between the R606 and the E605 would be also disrupted when R606 is symmetrically dimethylated, as methylation increases the bulk length of the side chain and also disrupt the hydrogen bonding capacity of arginine. So here is a model. So basically, the R606 residue by itself is involved in the interaction interface that is required for promoting RIPK1 autophosphorylation and activation. And the mutation of this, the dimethylation of this residue or mutation of this residue all disrupt this interaction, and this will lead into the inhibition of RIPK1 kinase. Okay, so using structure biology, we now explain how this residue and how this methylation of this residue regulate. So in line with this model, also decreased the self-association of RIPK1-DD, and we also generated the E605 the reconstituted cells, and these cells induced by methionine deprivation, responsible for catalyzing the dimethylation of the R606 residue. So arginine methylation is catalyzed by protein arginine methyltransferase, or PRMT, and the Mammonia PRMT family consists of line members, but we found that only PRMT5 deficiency sensitizes the cells to TNF-induced cell death that is dependent on RIPK1 kinase activity, because the RIPK1 kinase inhibitor like one can inhibit the cell death, but not the cell death in other PRMT deficient cells. Okay, so this result suggests that PRMT5 is a potential or the major enzyme that catalyzes RIPK1 dimethylation. And consistently, we found that TNF-alone can induce RIPK1 activation as marked by the S166 phosphorylation and S cleavage in PRMT5 deficient cells, but not in wild type cells. Just as we said before, TNF-alpha alone is not sufficient to induce cell death in wild type cells, right? And this effect were all inhibited by RIPK1 kinase inhibitor, LIK1. So we next perform an in vitro methylation assay in which the dimethylation of recombinant RIPK1-DD protein was only detected in the presence of PRMT5 and SAM, and in vitro, and which was abolished by the R606K mutation. Okay, so this suggests that RIPK1 is a direct substrate of PRMT5, and the R606 is the major residue. So with in methionine sufficient cells, the R606 dimethylation of endogenous RIPK1 was readily detectable, but absent in PRMT5 deficient cells. And the supplementation of STEM can restore the dimethylation in methionine stuffed wild type cells, but not in PRMT5 deficient cells, suggesting that PRMT5 is required for STEM-mediated R606 dimethylation in cells. So to determine whether RIPK1 catalyzes dimethylation of RIPK1 in vivo, we generated a peptide PRMT5 locale decree transgenic mice. Deletion can result in a dramatic decrease in RIPK1 dimethylation. So this result suggests that PRMT5 directly catalyzes R606 dimethylation of RIPK1 in physiological context. Okay. So next, we studied the physiological significance of this PRMT5 regulated RIPK1 methylation. And the conditional locale of PRMT5 in the liver can lead to spontaneous development of hepatocellular carcinoma, or HCC, as marked by the formation of tumors and increased Ki-67 positive proliferating cells in the liver. And strikingly, we found that the HCC development was largely inhibited by RIPK1 kinase dead mutation, suggesting that the abnormal RIPK1 kinase activation promote HCC development in PMD5 positive cells in the liver, in PMD5 deficient liver, marked by the increased serum levels of ALT and AST in PMD5 deficient mice. And again, these pathologies were all inhibited by RIPK1 kinase dead mutation. Okay. So next, we want to see how much the kinase activation of RIPK1 contribute to the pathologies induced by PMD5. So we performed the RNA-seq analysis. So basically, we found that more than half of the genes that were upregulated by PMD5 deficiency can be restored to almost normal levels by RIPK1 kinase dead mutation. So these results suggest that maybe we know that PMD5 have many substrate, and perhaps RIPK1 contribute to half of the function of PMD5. Okay, because more than half of the genes can be rescued, right? So, and again, these rescued genes are all involved in inflammation and cell death, apoptosis. So this result suggests that PMD5 deletion induces RIPK1 mediated liver damage and inflammation, which may contribute to the development of HCC. So because the increased inflammation in the liver may promote steatosis, and indeed we found that PMD5 deficient mice displayed steatosis as determined by all red O staining, which was also inhibited by RIPK1 in RIPK1 mutant mice. And we noted that PMD5 deficient mice also displayed a very strong fibrosis, which was confirmed by the Mason's trichrome staining that detect the collagen deposition. And notably, these fibrogenic changes was significantly reduced in RIPK1 mutant mice. Okay. So finally, because it is known that PMD5, we found that this PMD5 liver-specific lockout mice have a reduced lifespan, but RIPK1 kinase dead can substantially extend their lifespan. So again, even though PMD5 may have many other substrates as reported by many studies, we provide genetic evidence to show that RIPK1 is a very important physiological substrate of PMD5. Okay, that is required for maintaining at least liver homeostasis. Okay, so here is a working model. So under some sufficient conditions, when methionine metabolism and 1-carbon metabolism are normal, some abundance is sensed by RIPK1, where PMD5 can mediate the dimethylation of RIPK1 at R606 to inhibit its activation. And some sufficiency can also lead to the TNF promoter hypermethylation, which suppresses the transcription activation of TNF. And in contrast, SAM deficiency, first, it can induce the hypomethylation of TNF promoter, and this will induce the initial autocrine TNF production that is required for the downstream pathway. And the SAM deficiency also leads to the loss of RIPK1 dimethylation, and this unmethylated RIPK1 can undergo R606 residue-mediated dimerization, and upon the TNF detection, and this triggers RIPK1 kinase activation, and then triggers apoptosis and inflammation, and which may contribute to many pathologies such as HCC development. Okay, so basically this work is mainly done by my PhD student Chen Zezhao, and this project was also conducted with the help from our collaborators from in our research center, including Professor Dr. Zhang Yixiao, Dr. Zhang Yixiao for the cranium structure, Dr. Liu Cong also for the cranium structure, and Dr. San Bin for the mass spectrometry analysis of the methylation site, and Dr. Liu Jianping for the protein purification, and Dr. Zhu Zhenjiang for the metabolite mass spectrometry analysis of the metabolite, and Dr. Chai Yuping also for metabolite study. And okay, I will stop here, and I'm happy to take any questions. Thank you.